Sometimes the things we think we understand best, the really basic stuff, they can hold the biggest surprises, like how molecules move around in the liquid. Seems simple, right? We've had a model for that for ages, but well, what if I told you a new discovery is kind of turning that whole idea on its head? Today, we're doing a deep dive into exactly that, a totally new kind of molecular motion. They've seen it in these DNA polymer droplets. So our mission here is to unpack why this is so unexpected, how it actually works, and maybe most importantly, what it could mean for, well, everything from our own cells to maybe even future medical treatments. And this isn't just some small adjustment, it's a major rethink. The research comes from some really top places: Johannes Gutenberg University Mainz, the Max Planck Institute for Polymer Research, UT Austin. And they've built so much on the idea of random diffusion. It's like textbook stuff. So when Wexcan Chen, one of the key researchers at JGU, says, and I quote, "This is an effect we did not expect at all." I mean, that really tells you something. It's not just surprise, it's fundamental. Okay, so let's really get into this. How does it work? We all picture it, right? Molecular movement in liquids. It's that random spread simple diffusion. You put a drop of dye in water, it spreads out. Soft blurry edges, molecules just bouncing around.
Exactly. That's the classic picture. Random, chaotic. But what they saw here is completely different. In these special DNA droplets, the guest molecules, they don't just diffuse out. No, they move in what they describe as a clearly defined frontal wave.
Full wave.
Yeah, like an organized front moving through the droplet, not a random cloud. Professor Andreas Walser from JGU, who led the project, he put it this way: the molecules move in a structured and controlled manner, contrary to the traditional models. And this takes the form of what appears to be a wave of molecules or a mobile boundary. It's ordered, almost, directed. Totally unlike the chaos we expected.
Wow, okay. So not like people wandering randomly in a crowd, more like they're forming a line or maybe even doing some kind of coordinated dance. That's a huge shift. What's making them line up like that? What's the mechanism?
Right, so the setup is key. They use synthetic droplets. These are made of thousands of DNA strands. Think of them as biomolecular condensates. You find similar things in our cells, little compartments without membranes, organizing things.
Like little molecular meeting rooms.
Kind of, yeah. They concentrate molecules for specific jobs. And the great thing about these synthetic ones is you can tweak them. You can change the DNA sequences, change the salt concentration, fine-tune their properties. Chen actually pointed this out. He said, our synthetic droplets represent an excellent model system to simulate natural processes and understand them better.
Okay, so they got these controllable droplets. What did they add to see this wave thing happen?
They added specially designed guest DNA strands. And here's the clever bit, the key and lock mechanism.
Yeah.
These guest strands are designed to recognize and bind to the DNA inside the droplet.
Okay, they lock in.
Exactly. And when they bind, something really interesting happens. They actually make the material right around them less dense. So imagine the droplet is like a thick gel. Where these guest molecules bind, it locally becomes, well, more fluid, looser.
So it makes it easier for the next bit of the wave to push through. It sort of clears the path.
Precisely. It reduces the drag. Chen described the wave moving forward in a linear fashion over time. He said it's driven by chemical binding, material conversion, and programmable DNA interactions. And then he added, this is something that is completely new when it comes to soft matter.
Soft matter, like gels, liquids, biological stuff.
Exactly, things that deform easily. Seeing this kind of organized linear wave in that context, it's apparently unprecedented.
That is fascinating. It's like a chain reaction, almost self-propelling. So, okay, amazing science, but what does it mean? What are the implications here?
Well, on a fundamental level, it gives us huge insights into the physics of soft matter, obviously. But maybe more excitingly, it could really help us understand how our own cells manage signals and organize everything. Professor Walther thinks this discovery, it might be one of the missing pieces of the puzzle for understanding how cells regulate things at the molecular level.
Like finding a new rule for how traffic flows that suddenly makes the whole city's logistics make sense.
That's a good analogy, yeah.
So thinking about real-world stuff,
yeah,
what could this lead to? You mentioned medicine earlier. I mean, the paper talks about intelligent biomaterials, novel membrane technologies, even programmable drug delivery systems. That sounds incredibly futuristic.
It does open up those possibilities. Imagine drugs delivered not just randomly, but in a controlled wave to a specific target.
And synthetic cells that mimic biological organization.
Wow.
Yeah.
But how easy is it to go from these controlled synthetic droplets to, the messy reality inside a living thing? Is that a big leap?
Oh, absolutely. That's a huge challenge, bridging that gap. These synthetic systems are clean, controllable models. Real biology is, well, much more complex.
Right.
However, there's a really compelling potential link to neurodegenerative diseases.
Oh, so?
Well, in conditions like Alzheimer's or Parkinson's, you see proteins moving out of the cell nucleus and forming these condensates in the main cell body. And critically, these condensates can sort of age. They go from being dynamic and fluid to more stable, even solid-like. And that's when they often start forming harmful fibrils, those tangled protein clumps. Ah, I see. So this wave mechanism might relate to how those condensates form or change.
It's possible. Understanding this controlled movement, this transition in material properties driven by binding, it might give us clues. Walther actually expressed some hope here. He said, "It is quite conceivable that we may be able to find a way of influencing these aging processes so that over the long term, an entirely new approach to the treatment of neurodegenerative diseases could emerge."
That would be incredible. A completely new angle on tackling those diseases. So if we boil it down, the real aha moment here is that molecular movement isn't always just random bouncing. It can be this structured, wave-like process, at least in these specific systems.
Yeah.
And that fundamentally challenges our old picture. It opens doors for new materials, maybe much smarter drug delivery, and potentially even helps us understand devastating diseases.
Exactly. And it makes you wonder, doesn't it? If something this basic, this fundamental, can behave so unexpectedly, what other processes in biology or material science have these kinds of hidden structured dynamics that we just haven't seen yet? It encourages you to think about what might be happening, just beneath the surface of what we think we know, influencing our health, enabling new technologies. It really shows that challenging those core assumptions can lead to some truly massive breakthroughs.
It's a really powerful thought and kind of humbling. The universe, even at these tiny scales, just keeps surprising us with its complexity, its elegance, really.
Yeah.